in the Tropic Safe project, we are interested in the tolerance of citrus to HLB. And most of the investigation we perform are on polyploids, citrus polyploids. I will go fast on this second slide that is about the pathosystem. We know everything, the name of Huanglongbing, citrus graining, the three kind of bacterial species, Candidatus Liberibacter, the vector also. And in our case, we are more interested in the host plants. You have to know that all the citrus species and also some wild rutaceae are infected by HLB. And there is no variety, no rootstock that are resistant to HLB among the grown citrus. There is only a few related citrus that are resistant. You have to know also that the environment is very important. You have an effect of water deficit, other disease like Phytophthora or citri that impact the trees that are infected by HLB. So what is the dynamic of in plants and what are the physiological impact of HLB disease? When the psyllid is going to the tree, it will feed to the young shoots, and it will transmit the bacteria to the plants. So you will have a reaction of the tree, and will mean that you have in the phloem cells, you have that on the right of the screen that are alive, you have synthesis of callose in the phloem cells that will lead to the clogging of the pore that you can see here that put in contact all the cells. So what you will have, you will have accumulation of starch and sugars in the leaves. That is the reason of the symptoms that you can see here, because you have a perturbation of the metabolism. So the tree, the roots and the fruit are no longer supplied with this, not supplied with the elaborated sap, and the root system at the end is destroyed. So in our work, we are interested to look for better tolerant rootstock and science that can limit the impact of the bacteria. You can see the impact in Guadeloupe. I will go very fast. Yuri said a lot about that, and you can see that we got a huge drop of the production when the disease arrived in 2012. You can see that the only variety that was less affected was the lime, the Tahiti lime, or second name, the Persian lime, that is triploid, a natural triploid. We are interested by polyploids, so triploid and tetraploid, because they are specific interest. Most of the citrus are diploid, and you can see the impact of the polyploidy when you are tetraploid on the seeds. You can see the seeds are bigger, the cells are also bigger. You can see that on the picture number B, you can see that the tetraploid are smaller and greener than the diploid. You can see also that organs, the stem and the roots are bigger in tetraploid compared to diploid. You have also impact at the leaf and also the fruit level. And since we show that in the field, the triploids lime were less affected, we are wondering what was the reason. And I will present you some of the data we obtained and that are now published in Frontier in Plant Science. So we worked on Mexican lime and Persian lime, so the Tahiti lime, comparison between diploid and triploid. You can see the leaves on the left, the control that are green. And we worked also on asymptomatic leaves and symptomatic leaves. And you can see that the yellowing that is due to the disease. So what are the triploids better tolerant? The first thing we did was to look for the charge of bacteria. And you can see that in the control here, there is no bacteria as expected. But in the asymptomatic Mexican lime and Persian lime, and also symptomatic leaf, you can see that there is amplification of the bacteria. And the amplification of the bacteria in the asymptomatic leaves is lower. You have a higher CT in the triploid. So Persian lime is less affected when at asymptomatic level. We also looked at the microscopic level by electronic microscopy what is happening. And here are pictures of the cells of the phloem in the peciole, in diploid and triploid. Here you have the control. You can see that the cells are bigger in triploid compared to diploid. You can see the holes that put in contact the cells, and these holes are bigger in triploid. So you may expect that the flux of sieve is higher in the triploid. You can see that in asymptomatic leaves, everything is plugged in the diploid. There is no possibility of the sieve to go from cell to cell, and there is accumulation of starch, which is not the case in the triploids, where you can see that still the holes are open and the flow shape can continue. In symptomatic leaves that are dramatically impacted by the disease, is plugged and there is no transport of sieve anymore. This is also confirmed by results we did by using aniline blue. That is a way to estimate the presence of callose. In control, there is no presence of callose in the control, but you can see that in asymptomatic Mexican limes, there is a white ring that is showing the presence of callose that is not present in the Persian limes. And in the symptomatic leaves, of course, the presence of callose is present in both the case. So we confirm that in asymptomatic leaves, the triploids react better. So we did other things by measuring the organ of the size of the organs, the pores, and you can see, as you can see here, that the size of the peciole is much bigger in the Persian lime. This was expected compared to the diploid, and also interestingly that the size of the pores are much bigger in the triploid. And in asymptomatic leaves, it's still possible to measure the size of the pores. They are still open. Regarding presence of starch in the leaves, in control, you can see that there is no presence of starch. This was expected because all the starch are transported to the roots and the fruits. And you can see that in the symptomatic leaves, you have a huge accumulation, 10 times higher in diploid and triploid. But in asymptomatic leaves, the accumulation is much higher in the diploid compared to the triploid, showing that the triploid is able to translocate this starch to the leaf, to the other organs. We evaluated also other enzymes that are involved in the detoxification of the plants. We evaluated MDA, that is a markers of the stress, and you can see the stress is much higher in the diploid, as expected. We also measured that enzyme involved in the detoxification are more effective in the triploid compared to the diploid, and also for antioxidant enzymes. So as a conclusion of results, you can see on this drawing that you have a bigger cells in the triploid and bigger holes in between cells that allow to maintain higher flux of sieve in the triploid compared to the diploid. When you look for asymptomatic leaves, you can see that still you maintain the flux of sieve in the leaves, allowing the plant to be in better shape, and also you have better detoxification, photosynthesis that is higher. So this together confirm the better tolerance of the triploid. In symptomatic leaves, all the leaves are affected, and in that case, you have only a better flux of the sieve in the triploid compared to the diploid. Thank you very much for your attention, and I will reply to the question if you wish. Thank you.